So what I was doing was looking at Andalithic biodiversity of Earth and a terrestrial Mars analog. So Andalids are basically organisms that live inside of rocks. I don't know if you can see that in there. But
let me zoom in.
But there's a green layer right along the bottom of that rock there, or right on the inside surface. And that's actually cyanobacteria that have colonized inside of this rock, and they're photosynthetic. They actually use wavelengths of light that can penetrate just the top surface of the rock. So what I'm looking at these for is as a potential model for life on Mars. So we know the surface of Mars gets really cold, there's not much pressure, lots of radiation. It's really not a good place to be if you're a microbe or anything. But if you move into the subsurface, things get warmer, there's moisture there. So we're more likely to find organisms on Mars within the subsurface or in these kinds of environments. So what I was doing was I went to the Mars Desert Research Station at the URCAD Desert. So it's a Mars analog station. And so what we do is volunteers actually come for 2 weeks here in a Mars simulation. So this is me in a spacesuit collecting rock samples. What I've done was pick several sites surrounding this habitat. So they keep environments that seem to have evidence of past water. So dry stream valleys, sandstone deposits, all kinds of environments that seem to have had water in their past, and also had this evidence of colonization inside of it. So I brought all of these samples back to UMBC for analysis. So the overall goal was to determine the biodiversity of these. So one of the things that I had done was some fluorescent microscopy to determine that these green layers are in fact organisms and not some kind of mineral deposit. So these top two are controls, so looking at areas that appear to be uncolonized. You see in each different, these are all 3 different types of fluorescence that I looked at, and they're lighting up in about the same spots with about the same patterns. As compared to here, these are both colonized regions where you see these green layers, and they're lighting up a lot brighter than this red fluorescence versus the other 2 kinds that I looked at. And it's on the same pattern, it's just cyanobacteria suspended in water. So they only lit up under this red fluorescence. So taking this as the indication that there's probably some sort of cyanobacteria inside of these samples. What I also done was extract DNA out of these and amplify the 16S rRNA genes. So that's very useful for species classification. Every species has its own distinct 16S sequence. So it's good for any kind of phylogenetic analysis. So we did with those, so this here is a denaturing gradient gel electrophoresis. And so what this does is takes those 16S sequences that have been amplified from each one of these sample sites and separates them based on size and charge. And so each band here is a different species. So what this is showing really, this is very new results, I got it just yesterday. It's very exciting. And what this is showing is that, first of all, this very complex communities inside of the samples of these rocks in an environment that looks like it doesn't have any life at all. Just look at it basically. There's a lot of species in here, a lot of different diversity, and there are differences between the different sample sites and the different rock types that I'm looking at. So hopefully I will have time to look at a couple more different samples, do the same thing, make the gradient on this a bit smaller so you can zoom in, spread these bands out, and really see where things are lining up.